Thank you very much for giving me the opportunity to speak, and I'd like to, of course, thank the Children's Cancer Foundation for supporting work I've done for so many years. I wanted today to give a bit of an overview of the kind of work that CCF has supported, and really they started supporting me 18 years ago now when I was still a clinical fellow working with Peter Berger. So I'm going to do a bit of a overview then of the kinds of studies and the impact they've helped us do over the last 18 years and how some of those things have progressed. Is it? Yeah. Whichever. It's. Okay. I can get that if you can get the clickable work. In children, and currently the standard of care is radiation and chemotherapy, but there's been a major focus over the last 20 years or so to come up with more targeted therapies that are more specific for different subsets of medulloblastoma, and also finding ways to reduce therapy in children who might have less aggressive forms of medulloblastoma, because as many of us, or almost all of us in this room know, there's a lot of side effects from these therapies, and if we could reduce them, that'd be a great thing. So, as I mentioned earlier, really this goes back quite a while for me, all the way back to 1998 when I was just starting my clinical training at Johns Hopkins. And one of the first studies I worked on was with Peter Berger, who had funding at the time from the Children's Cancer Foundation. And what we had looked at was the use of beta-catenin immunohistochemistry to define a subgroup of medullos that had activation of this so-called Wnt signaling pathway. And that was important, it turned out, because it helped to define a subset of cases that was shown several years later in a study done in the United Kingdom of a clinical trial, where those tumors that had this nuclear translocation of beta-catenin turned out to be in children who did much better than the average children with medulloblastoma. And based on that now, there are clinical trials that have been begun in the United States in which they're reducing the therapies for these children in the hopes that you can get away with similar very good long-term outcomes, but with less bad side effects of the radiation and chemotherapy. So this is one example of how we're trying to understand the biology of the tumor in ways where you not just try different kinds of therapy, but in some cases maybe we just need less therapy and we can have equivalently good outcomes. So I'm a neuropathologist. I look at these tumors under the microscope and decide what the diagnosis is. And for medullo, they can look a lot of different ways. So shown here are three of the main histologic patterns. You can have so-called classic medulloblastoma, a large cell anaplastic medulloblastoma, nodular desmoplastic medulloblastoma. And one of the next things I worked on was with Peter Berger again, looking at this large cell anaplastic medulloblastoma subtype, which really just looks nastier and more aggressive than your average medulloblastoma. And what we did then, again with funding from CCF, was to look at grades of anaplasia. So you could have only slight anaplasia, moderate or severe anaplasia, and again looking at a correlation with outcome. Looking at a correlation with outcome cases, and this is now a retrospective analysis of clinical trials data from pediatric oncology group, those with the patients who had severe anaplesia in their tumors had only about a 20% long-term survival, whereas the majority of patients who had no anaplesia or slight anaplesia had a 50% or more long-term survival. So now we're defining not a subset that have good outcomes, but a subset that have bad outcomes. As I'll tell you in a second, I went on to look at the molecular drivers of this, but this is just, throughout this, I'm going to try to highlight some of the papers we published along the way with support from CCF. So these are the first 3 papers that we published with support from CCF. So around this time, I was joining the faculty myself, and I wrote my own first grant to CCF under my own name to look at the role of an oncogene known as MYC in the aggressive large cell anaplastic medulloblastoma. This is just showing how normally there would be 2 copies of the gene, these red dots in the panel labeled normal, whereas this gene becomes amplified in the more aggressive tumors, and you can see 50 or more copies of that. And it turns out that these histologic subtypes of medullo have some correlates in terms of the molecular pathways that are turned on in them. And it's the large cell anaplastic aggressive tumors that tend to have high levels of MYC. And we now know that you can separate medullos into 4 main molecular subgroups. A lot of the neuro-oncologists in the room will be tired of seeing this diagram. There's a Wnt group, a sonic hedgehog group, a so-called group 3 that tends to be the large cell anaplastic tumors that have MYC amplicons, and group 4 that's also has sort of intermediate outcomes and is poorly understood. And so these groups that were initially defined by expression profiling have recently been incorporated into a new WHO classification scheme that just came out this month, actually. So here was the group that met in Heidelberg last summer to finalize the new WHO brain tumor classification scheme. And here is the, can we go back one? Okay, I can't get it to reverse. Yeah, the back won't work. Thank you. Here's the new, for the neuro-oncologists and oncologists in the crowd, here's the new WHO classification scheme that was just published this week. And for the medulloblastoma group and for a lot of these brain tumor groups, we now have incorporated molecular diagnostics into the definition of these tumors. So there's a genetically defined set of medulloblastoma subgroups, a Wnt group, a sonic hedgehog group, which can be either p53 mutated, which sadly have very bad outcomes, or p53 wild type that tend to have pretty good outcomes, and then these non-Wnt, non-hedgehog tumors that fall into groups 3 and group 4. So we're moving forward. It turns out that these are very different tumors in terms of the types of children they arise in, the outcomes, the kinds of therapy we should be using. And so this classification is really now integral to how we're going to be diagnosing these tumors moving forward. So there's been real progress, I would say, over the 20 years I've been working on medulloblastoma in terms of understanding the disease. Now, of course, understanding the disease isn't enough. We want to come up with better therapies for the disease. And in this first grant that I got myself from the CCF, we tried to model these high-risk medullos by taking lines that had low levels of MYC and increasing MYC and seeing how that affected the tumor biology. And the tumors became much more aggressive in how they grew both in vitro and in vivo when we increased the MYC levels. And we published this a number of 10 years ago now in cancer research, but we're still using these lines that were generated with CCF funding 10 years ago. In fact, we had a paper that came out in Clinical Cancer Research last month, which we're still using these paired lines with high and low MYC levels. So again, when you invest in research tools, these things can yield dividends over very long periods of time. Here's just some of the papers that came from that first grant I got from CCF under my own name. Now, and I should say that with the preliminary data, one of the things I'd like to emphasize both to the academics here, but also to the patient groups and the people who are helping to raise money. At the time when I was just getting going, even a very small amount of seed funding can have a huge impact, because basically that preliminary data I generated looking at MYC, I then was able to go to the National Institutes of Health, the NIH, and get my first career award grant that supported my laboratory for the first 5 years it was in business, a so-called K08 award. So one of the things that CCF does that's wonderful, and I think many of us here in this room have benefited in this way, where you get early career funding that gets the preliminary data you need to get larger grants from the government. So the next thing we did looking at medulloblastoma was addressing the issue of cancer stem cells. And about 10 years ago, people became interested in the notion that it wasn't every single cell within a tumor didn't behave the same way, and that there were stem-like cells sort of at the apex of a developmental hierarchy in the cancer. And those stem cells are really the only cells in the cancer that could divide forever. And that if you killed the rest of the cancer, and the rest of the cancer is more sensitive to standard radiation and chemo, whereas these red cancer stem cells are more resistant to standard radiation and chemo. And if you don't kill these stem-like cancer cells, the tumor's ultimately just going to grow back. So this was a popular idea. I think there's a lot of truth to the idea, although some of the specifics haven't held up to the test of time. The general idea that there's heterogeneity within a tumor and different therapies may be necessary to target different subsets of cells in that tumor, I think most people believe to be true. And so the next thing we were working on then in my laboratory was trying to understand how we can kill these stem-like cancer cells. And I turned to the analogy or the notion that the pathways that drive the normal development of the brain probably were similar to the pathways that were involved in these cancers. So if you think how rapidly the brain develops during fetal development and early childhood life, and if you think of the cellular processes that are going on during that time, they're very similar to what's happening in cancer. And so we looked at whether the same pathways that were important in normal development, the Wnt pathway, hedgehog pathway, Notch pathway. A lot of these pathways have funny names because they were discovered by fruit fly biologists, and they like to give interesting names to things. We looked at whether these pathways were important. And so one of the pathways I focused on a lot is Notch. It was discovered 100 years ago in flies because they got notches in their wings. That's where that name comes from. And we found that there was a stem-like population in these medullos. When we inhibited Notch, we could get rid of this stem-like population, and that had a profound effect on the growth of the tumor in vitro and in vivo. And in fact, in the interest of time, I'm not going to go into a lot of the research details, but ultimately, these Notch-inhibiting drugs were tried in both adults and children with brain tumors, and they're still working through some of the issues. That it hasn't been a home run in terms of its efficacy, but there's some hints that Notch inhibition will be useful in a subset of these cancers, particularly in combination with other kinds of therapy. And we're continuing to look at Notch as a potential therapy for a wide range of cancer here, turning from medulloblastoma and so-called embryonal brain tumors to astrocytic tumors. Here's a paper that was just published from our group with collaborators, Javad Nazari and others from this area down in DC, looking at DIPG, diffuse intrinsic pontine glioma, brainstem astrocytic tumors similar to the ones we just heard about from Mrs. Offit. And here we have a paper that was published last year in which we think that maybe inhibiting the Notch pathway can be effective in treating children with these devastating brainstem astrocytic tumors. So around that time, there was a group of patients who were interested in learning more about pilocytic astrocytoma. At the time, there was not a lot of work being done on low-grade astrocytic tumors. And so we started working on those, and we'd sent a grant to CCF that got funded to look at the genetics of pilocytic astrocytoma, which of course are the most common low-grade tumors in children in the brain. And around the same time, 9 years ago or so, we found in my laboratory and a number of other folks around the world, basically all discovered within a period of 12 months of one another, that the majority of these low-grade astrocytic tumors have changes that affect the BRAF oncogene on chromosome 7. And this is just some of the primary data from the paper we published then describing our results. And it turns out that what happens is there's a duplication of about a 2 megabase region on that chromosome that results in fusion of the business end, the active kinase domain of BRAF, to this KIA 15549 protein, and that results in unregulated BRAF kinase activity. And so I showed you initially a picture of a pilocytic astrocytoma of the optic nerve, but really it's pilocytic astrocytomas all through the brain that have these changes, although they're most frequent in the posterior fossa and brain stem, as well as the optic nerve. Now, importantly, this tells us something about the biology of these tumors that, again, can result in improved therapies. So we know that there are at least two arms to the signaling pathways downstream of the RAF signaling, and really both of these arms have drugs that can target them, and both of these arms have been targeted now in clinical trials in children. Our initial attempts to target the BRAF kinase and the downstream MEK-ERK signaling weren't so successful. We think we have a better understanding now in collaboration with folks at CHOP as to why that might be. There's been more success in targeting the mTOR signaling pathway, but this is still a very active area of investigation in our laboratory. Now, one of the problems with working on the low-grade astrocytic tumors is a lack of good models, and working with Eric Raab, we published a paper a few years ago in which we used human neural stem cells to try to model these tumors. And we found that one of the reasons that we've had a hard time growing cell lines from these low-grade astrocytic tumors is a process known as oncogene-induced senescence. And it turns out that P16 is a key player in driving this oncogene-induced senescence, and it's also associated with worse outcomes in patients. So from that paper, here's some survival data on children who have these tumors, and if you lose P16, your tumor doesn't senesce, it doesn't stop growing, and there's a much worse survival in children who have P16 deletion and protein loss as compared to those who retain that oncogene. But we're hoping to use this increased understanding as a way to grow these cells in culture. We're also trying some other tricks, because one of the things that's kept us from coming up with better therapies for this tumor is a lack of good models, culture models that we can use to grow them. So we've been trying to develop culture models both in cultures and in mice. And some of the hints to what's going on with the biology of these low-grade astrocytic tumors and optic nerve astrocytic tumors in particular came from work that we had done in collaboration with an investigator known as Nick Gaiano. And working together, we had done some gain-of-function models in which we had, these are amazing experiments that Nick used to do, where basically, shown in the upper left here, is a mouse, and the uterus is surgically exposed. It's a pregnant mouse, and this is an ultrasound probe, so you can visualize the fetuses within the womb. And so here's a single fetus, and this sort of kidney-shaped thing on the right is the brain, and you can actually, under ultrasound guidance, stick a needle into the brain of this mouse fetus in utero, and inject it with retrovirus, encoding, in this case, activated Notch3. And we had done those experiments initially and shown that could drive chorioplexus tumor formation. And subsequently, when we kept following larger cohorts of animals, we found about 50% of them also got glial tumors involving the optic nerve that looked a lot like the optic nerve pilocytic astrocytomas. And so this is another model that we've been working with over the years, looking at pathways important in pilocytic astrocytoma. And here's a paper we just published last year, our group, looking using some of the rare cell lines that exist for this, again, focusing on Notch inhibition as a possible way of targeting these low-grade astrocytomas and showing that we can inhibit growth of these cancers genetically by inhibiting Notch. So there's just some more papers that we'd published. I think this was, I stopped listing them. This was 12 papers funded by the CCF up to 2012. The most recent grant I had from CCF actually was focusing on higher grade pediatric astrocytic tumors and looking at a pathway of DNA telomere maintenance that's common in pediatric high grade astrocytic tumors. So, to maintain your telomeres, of course, when cancer cells grow, often that's done by turning on the enzyme telomerase, but it turns out in pediatric astrocytomas, more frequently what happens is there's this pathway known as alternative lengthening of telomeres, or ALT, that goes on. And researchers at Johns Hopkins, Alan Meeker's group, had shown that ATRX mutations are key drivers of this ALT pathway in gliomas. And we had generated from a patient a cell line that had this ATRX loss and alt phenotype, these big pink blobs in the cells are telomere signals, and normally they should be too small, a few small dots in each cell. So the fact that you're seeing a subset of cells with these big blobs in them is indicative of the fact that there's alt going on in this cell line and in the primary tumor from which it derived. Now, interestingly, the cell line didn't have complete loss of ATRX. There's only partial loss of ATRX. So we've been using it as a model to try to understand what happens when you start with partial loss of ATRX and knock out the rest of it. So we use genetic tricks to knock out, completely get rid of the last ATRX expression. This is shown here on the left panels using 2 different hairpins. We got rid of it. And the effect of getting rid of ATRX completely in these cells was that you get a very significant induction in clonogenic growth, shown on the panels on the right. This also causes a really significant increase in the growth both in flank xenografts, again shown on right, where you're measuring week over week, and the red lines are individual flank xenografts growing much more rapidly in the tumors in which we've knocked down ATRX as compared to the vector controls shown in blue. And then we also have done intracranial xenografts. So we're continuing to work on this as a way of trying to understand what the effects are of ATRX knockout in these tumors. And this was work that had been funded by CCF. That grant finished up a year or 2 ago. And we're still, though, getting the study wrapped up and published. So this is unpublished data. Perhaps most important, though, in this unpublished data I'm showing you, is not the fact that knocking it down makes it grow faster, but that knocking it down seemed to promote a resistance to standard chemotherapy, standard chemotherapy such as temozolomide. So this now is telling us something about the biology of how ATRX expression may modulate response to standard chemo, and we think we understand why this is happening at the level of DNA repair and how we might get around it in terms of treating children who have these kinds of cancers. So I apologize for going really rapidly. There's just been so many things that the CCF had supported in my group over the years. I wanted to give people a flavor for the broad spectrum of work that we had done. I should add that in addition to the K08 award that had come from CCF, we got an R01 grant funded based on preliminary data that they had supported, and that got that R01 renewed about 4 years ago. So I've had really about 12 years of uninterrupted support from NIH, and in almost all of those grants that I got, the initial seed data was funded by CCF. So it's really a way that CCF can leverage their funding to get larger amounts of money from the government to keep working on these studies. So thank you very much.
Thank you. That was really nice show of how a little bit of investment can really pay dividends. Huh?